Hello biology students. Today I'm going to be writing a DNA molecule, taking that DNA molecule, writing a messenger RNA molecule from it, and then writing a polypeptide chain from that messenger RNA. So let's start here with DNA, and we need to write one side of the DNA strand. Okay, so there's one side of the sugar phosphate backbone, and let's just write some base pairs. We have an adenine, we have a thymine, guanine, maybe another guanine. Alright, maybe cytosine. Let's see here, adenine, and then maybe cytosine. Alright. Now, of course, we need to show complementary base pairing, and so adenine A always bonds with thymine, which is a T. We'll go down through here. Here's two more adenines, so they're going to bond with thymine. Now remember, we're talking about DNA here, so in DNA, we're talking about a thymine molecule, not uracil. Alright, now we'll go to the thymine here. It bonds with adenine. Okay, and then we have some guanines there. Guanine bonds with cytosine. And then we have some cytosines that bond with guanine. Alright, now we got the one half of our now add the sugar phosphate backbone. And there we go. We have a DNA molecule. Double helix showing complementary base pairing bonded with hydrogen bonds. A bonds with T, T bonds with A, G bonds with C, and C bonds with G. Now we can write a messenger RNA molecule from that. Okay, now remember, in biology, we're going to use one side as a template strand. So we're going to use this side right here as our template, okay? And we're going to use that to create our messenger RNA. Because remember, one side of the DNA is used as a template to read off of. And we're going to still use the same complementary base pairing rules, but now that we're talking about RNA molecule, we're going to be using uracil instead of thymine. So T will be bonding with A again. Oops, should be green. There we go. Now A is going to bond with uracil. Remember, it's uracil, not thymine. So there's a U. C is going to bond with G again. Same thing here. Now G is going to bond with C, which is cytosine. We have some thymine molecules. They're going to be bonded with A, which is adenine, and then guanine down there bonded to a cytosine. Alright, so there's our messenger RNA molecule. Very simple, guys. The only thing to remember is just making sure that you're using uracil instead of thymine. Okay? So now we can go on to create a polypeptide chain. Okay, so polypeptide chain. Now this is when you're going to need to Google an amino acid chart. Okay, because we're going to have to read the codons. Now if you remember in my class, codons are grouped in groups of 3. So you read them in pairs of 3. It's just like deciphering a code. So now we have 3 codons here. Remember, three base pairs make up a codon. So we have AUG, GCC, AAC. So then now you're going to use the amino acid chart that you just Googled to look at AUG and figure out what AUG is. And we look at AUG is the amino acid for methionine. Now remember, I'm going to do a video on tRNA. Transfer RNA is the one who brings the amino acids to the ribosome for translation. Now we have GCC. GCC, if I look at my amino acid chart, is alanine. So let me get another color out here. Draw my amino acid, ALA. I'm just going to abbreviate these. Now remember, these are bonded by peptide bonds. Peptide bonds. Alright, and lastly, AAC. AAC is asparagine. Now, you remember, once again, you find these on the amino acid chart. Just Google it. And I'm just going to write ASP for short. Draw my peptide bond. And there we go. There's a small polypeptide chain. If you some bonus points, why did I use AUG? Why did I use AUG? And AUG, remember, is the start codon. Tells it where to start. So AUG is the start codon, okay? Anything before that won't do anything, but as soon as you get to the AUG, it's the start codon, and things start happening. Alright guys, essentially that is the central dogma, going from DNA to messenger RNA being transcription, and then taking that messenger RNA and writing a polypeptide chain or a protein being translation. Alright guys, stay tuned for my video on transfer RNA, and I hope that helped.